A double strand of DNA unwinds, and the strands separate. For each unwound strand, the bases can now match with those that are floating free in the cell. The familiar base pairing rules apply. A pairs with T, G with C, and so on. So each strand of the double helix acts as a template for the formation of a new complementary strand. This eventually gives two DNA double helices, each identical to the original and actually containing one old strand and one new.
The blood group gene makes a protein. When this has to be manufactured, a copy of the gene is taken in the nucleus, and this messenger molecule carrying the code for the blood group protein travels out into the cell. It's made of RNA, like DNA, but it uses U instead of T. It turns out that the 4 letters are in words 3 letters long. Each word fits a special adapter carrying one of 20 protein subunits, the amino acids. So the point of the DNA code is to carry protein sequences. Once the blood group protein chain has been put together, it folds up. The 20 amino acids can be arranged in any order, making all the proteins in the living world. But tiny DNA spelling mistakes can have a huge impact. The code for the A and B blood group proteins is an identical 1062 letters long, but the B code has 7 different letters. These mutations change the meaning of the words, so the B protein gets 7 different amino acids. The O code is identical to the A code except for a single spelling mistake, but the effect is dramatic. This is a deletion, a missing letter. Because the code is read 3 letters at a time, all the words downstream are now out of place, making a very different protein. The blood groups show that there is no such thing as the human genome. Although some genes have to be exactly the same in all of us, or their proteins won't work at all, thousands of others are like the blood group gene and can have 3 or 4 or more varieties. Since we have 30,000 genes, that's an awful lot of variation, and helps to explain why each of us is unique.
To make a particular protein in the cell, the relevant gene is first switched on in the DNA. A working copy of the gene, called messenger RNA, is made. This copying process is called transcription. Next, the information in the messenger RNA is acted upon to produce a protein. This step To understand how a working copy of the gene is made, we need to be familiar with the structure of RNA. Unlike DNA, RNA is just a single strand of nucleotide units. In DNA, the sugar is deoxyribose. In RNA, it's ribose. As for the bases, although 3 are identical, adenine, guanine, and cytosine, the thymine in DNA is replaced by uracil in RNA. Uracil is very similar to thymine. It always pairs with adenine. That is, it obeys the same base pairing rules.
Okay, so specific base pairing has cropped up again. You've already seen how important it is in the structure of DNA and the replication of DNA. And now you'll see how vital it is in the production of messenger RNA in a process known as transcription.
When transcription starts, a small section of DNA is unwound. One of the two unwound strands acts as a template for making the message. The messenger RNA is built up, one nucleotide at a time, according to the familiar base pairing rules. A on the DNA pairs with U on the RNA, G pairs with C, T pairs with A, and so on. The result? A message with a base sequence complementary to the template strand of the DNA. This messenger RNA will eventually be used to direct the formation of a protein.
Well, that's just about it. We've looked at the structure of DNA, how DNA is replicated, and how the information in DNA is utilized to make proteins. But underneath all of that, I think there's one principle that really helps tie things together, and that's the principle of base pairing. We've seen how base pairing helps explain replication of DNA. It also helps explain transcription to make messenger RNA, and translation of messenger RNA to make proteins. And of course, base pairing is also important in explaining why the DNA molecule is a double helix. It's quite a remarkable molecule, and one that provides a wonderful example of the connection between structure and function. It's also a very clever solution to the three problems that we set earlier for inherited material: stability, accuracy of copying, and containing information. Clever? Well, if molecules did have brains, this one would be Einstein.